Good day, my name is Malvika Sudhakar and I'll be presenting my poster titled Pivot, Machine Learning Approach to Identify Personalized Driver Genes Using Multi-omic Data. So let us first see what are driver genes. Driver genes are those genes which, when altered, give the cell an added growth advantage and hence a normal cell turns into a cancer cell. Now, these driver genes can be classified or categorized into two different categories called as TSGs or tumor suppressor genes and oncogenes or OGs based on their functionality. Now, tumor suppressor gene is usually inactivated by an alteration and then loses its function and then becomes a driver gene. An oncogene, on the other hand, when it is altered, it gains a function, it is activated by the alteration and becomes a driver gene. Now, why do we need to identify personalized driver genes? So we know the tumors are heterogeneous. That means they vary not only between the two cancer types, but also between two individuals within the same cancer type. This is because genes function differently in different tumors. While a large number of cohort-based methods exist for identification of driver genes, these cannot identify rare driver events which are observed in only very few samples. So identification of personalized driver genes can help us identify rare driver genes, which can then be used for diagnosis and treatment. Now, the current methods which exist for identification of personalized driver genes are based on unsupervised methods. They use network information and are based on the principle that the neighborhood of the altered gene will show a perturbation in its expression. So what we were interested while developing Pivot is to generate a supervised method for identification of driver genes. So we basically pre-process the data into different kinds of features, which are then labeled, and we build models which are then validated, and based on the best models, we use them for predicting new personalized driver genes. Now, as I said, that we were interested in formulating a supervised problem, so we needed to label the data. Here we used 4 different methods for labeling. We used different data such as SNV, RNA, and multi-omic, different feature sets, and different ML-based algorithms for building the models. So we built models in all different combinations of labels, data, features, and models. So what we find is that among the labeling strategies, which can be classified into mutation-based strategies and gene-based strategies, mutation-based strategies are those where a particular known driver gene has a mutation in a known driver location, whereas a gene which has a mutation but not in a known driver location will be which will remain unlabeled. So, whereas in the gene-based labeling strategies, the location of the mutation is not really important, and any known driver gene which has any mutation is labeled as a TSG or OG. So what we find is when we look at the SNV models across the different cancer types, that the Bailey et al. Gene list helps build models that perform the best. This is because they not only can label across the different cancer types with unlike mutation-based strategies, they are also specific to the cancer type. The gene list is specific to the cancer type and hence outperform labeling strategies such as CGC. We then built SNV, RNA, multi-omic models, and we found that multi-omic models perform the best as they can label both mutation and CNV driver events. The classification metrics for the multi-omic models is given out here. We then looked at the number of genes that are identified in a given sample, and we find that around 4 to 5 genes are mutated for a given individual, which correlates with published data. The number of driver events, such as CNV, which were identified per individual, were far larger. We compared Pivot with other personalized driver gene identification tools, such as TransMed, and find that Pivot consistently performs better. We were then interested to see if the model is able to identify rare driver events. So we listed out all the genes which were frequently mutated, and we find that we identify known as well as new driver genes. We then mapped the number of samples each labeled gene is, each driver gene is labeled as. TSG and OG, and we find a large number of frequently mutated genes, but we also find a large number of genes which are mutated in very few samples, which is less than 10. And to see whether these are actually false positives or true driver genes, we looked at the distribution of known driver genes, and we find that the known driver genes has a similar bimodal distribution. Interestingly, we also find that genes do have dual roles within the same cancer type. Here I take the example of the JAK1 gene, which is identified as both a TSG and OG in different samples in BRCA and COAD. So JAK1 is a known signal transducer and is known to activate the JAK-STAT pathway. There are literature which shows that it needs to be consistently active in colon and breast cancers, which lead to lower survival rates, which shows the activity of oncogene. But there are also reports that say the JAK1 needs to be underexpressed if the cell needs to evade the immune response. So the role of JAK1 is highly dependent on the cell conditions in the microenvironment and varies across the different cancer subtypes. So to summarize, we were interested in developing a supervised problem, and we identified that the gene-based labeling strategy, which is cancer-specific, works the best. We built multiple models using mutation RNA and multi-omic data, and our models are able to label data as TSG and OG in individuals. They are able to identify rare, frequently altered genes, and also genes with dual roles in cancer types. Future directions for Pivot would be to extend it to other cancer types and also generate features which includes epigenetic data. Thank you. I would like if you would leave your comments and questions, and I'll get back to them as soon as possible.